Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome, everybody, back to In the Interim. I'm your host, Scott Berry. I have really honored to have my guest today, a very well-known award-winning statistician with me today. We're going to talk about a number of things with Dr. Senn. He's worked as a statistician, an academic in various positions in Switzerland, Scotland, England, Luxembourg. Has a really interesting history of work from being, which is a really cool title, the head of competence at the Center for Methodology and Statistics. I love that title. He's been a professor of statistics at the University of Glasgow, professor of pharmaceutical and health statistics at University College London. He spent 8 years at Ciba-Geigy as well. He's the author of Crossover Trials in Clinical Research, Statistical Issues in Drug Development, Dicing with Death. He was awarded the 2009 Bradford Hill Medal of the Royal Statistical Society, and 2017 the Fisher Memorial Lecture. And so I'm honored to have you on. Welcome to In the Interim, Stephen.
Okay, thanks for the invitation. The pleasure is mine.
So my father, Don Berry, has attributed a quote to you, and I'm wondering if you want to attach this. He said that you said, "I don't care whether someone is Bayesian or frequentist, as long as they understand the regression to the mean effect."
Yes, I tend to agree that. I often think that it's far more important to know how the data arrived. I think that the basic thing a statistician should always ask themselves, how do I get to see what I see? And the central fact about regression to the mean is that the data you're using as a comparator were selected to be what they are. And the data you're using to make the comparison are an outcome, and they're fundamentally different things. The patients only got into the particular trial because they had the values you defined. They wouldn't have got in if the blood pressure hadn't been above a certain level. After that, what you did was something different. You didn't set a criterion, you actually observed what happened. And that's basically, it's the asymmetry between those two things and understanding why this causes a particular problem. That sort of thing is a thing that statisticians ought to understand. If you can understand straight away why it's nonsense to claim that because actors who win an Oscar live longer, that the esteem of winning an Oscar is causing them to live longer, if you can see what the fundamental flaw is there, then you're beginning to be a statistician. It doesn't matter about being a Bayesian or a frequentist. Yeah, so...
Yeah. Well, and it may even come up as we talk about what is a placebo effect, which I think you laid out very much, which I think is very much misunderstood by many in clinical trials. So I want to talk. We want to talk, and you have been quite active on social media. By the way, I very much enjoyed the on Twitter your pictures of the various hikes you take. Beautiful hikes, which always seem to end with a picture of a beer. But those are fantastic. I hope you're able to still do those.
Yes.
Yep.
I hope to be doing a hike tomorrow. The weather looks good, so I think my wife and I will do a hike tomorrow.
Fantastic.
For Thursdays, I'm usually on my own because my wife has a walking club she's in, which I'm not allowed to join. So she goes off on a Thursday. I go do something else on the Thursday, and then other days we might be going out for a hike together. So.
That's wonderful. As part of Stephen's activity on social media, a number of really wonderful blogs. I suggest you read them. And then one of them came up, which is titled "Beware of the Morlocks." And loving Stephen's blogs, I go read it. It turns out maybe a criticism of something I was involved in. So we're going to get to that. But
Okay.
first, there's lots going on in the world here. The FDA guidance on draft guidance on Bayesian statistics. Part of your beware of the Morlocks talks about Bayes and not. I think it might be nice to throw it over to you about the Bayes, not Bayes, your position on that, and then maybe even leading into the FDA guidance and what you think of that.
Okay, so I can start with flexible designs. If you change the allocation ratio and you don't do nothing else about it, the net effect will be there will be an induced correlation between time and the treatment. Because at a particular era, more of A was given than B, and another era, more of B was given than A, or whatever. And so basically, unless you do something about it, time is confounded with the treatment given. And so a natural thing is to try and deconfound it. So the classical frequentist way is to say every time you change the allocation ratio, you declare a stratum, and you then essentially fit the stratum as a fixed effect factor. And that will force the construction of all estimates on the basis of within stratum differences. The only way you can eliminate the stratum effect is by constructing a difference, first of all, and then everything basically boils down to weighted combinations of these within strata differences. So that's what happens. But of course, in doing that, you're using up a large number of degrees of freedom. And you may ask yourself, can time really be this complicated? And so the natural thing to do is to say, well, maybe I could use a rather simpler function, one with rather fewer parameters. And if you have fewer parameters, the penalty you will pay for loss of orthogonality will be rather less. There's always a penalty for any loss of orthogonality in any regression model. The penalty will be less. And a natural way to do this, a natural way to be flexible, is Bayesian. But I don't actually see the Bayesian part of it as being essential. Essentially, what you're doing is you're replacing a multi-parameter adjustment with something which uses rather fewer parameters. That's one way of putting it. So that's the first thing. The second thing is one has to be a little bit careful. There is a correlation between time and allocation, but time covers a whole host of things, and this is not always appreciated. So the danger is you concentrate on time as if it was something continuous. Some aspects of it are. It's reasonable to believe in some smoothness of time, but some things are not so continuous. And I gave some examples in the blog. So that's basically my summary of what's in the blog.
Oh, okay. So let's set up the blog and do that. But before, I wonder if before we do that, just the Bayesian and frequentist thing. Given the quote that about regression to the mean, you're somewhat agnostic to this, and this is not anti-Bayesian. This is not anti-frequentist. This is about functionality of the model.
Sure. If what you did was you had an uninformative prior on each of these stratum effects, you would effectively be fitting a fixed effect frequentist model. It's only because you do something like you either have a polynomial function in which you do something like you penalize higher order terms, you make them less likely to be as large as lower order terms, or you imagine that you have somehow some pseudo data that you can add in some particular way to help with the particular adjustment. It's only if you start doing that, which is a perfectly natural thing to do for anybody modeling, then that then arises. What I think is dangerous if people say, oh, because I'm using Bayes, the problem is solved. There are still very, very hard decisions to be made in principle about how are you going to use Bayes. That's the big issue, not whether.
Okay, so let's set up the problem and maybe take one step back. Just if you read Stephen's blog, Beware the Morlocks, you can get there on his LinkedIn page. But to set up the problem is, and you discuss the Seville paper, which I'm a co-author on, that talks about the Bayesian time machine.
Yeah.
And so a scenario I think that we can use is that you have a platform trial, and the platform trial starts with a common control, and you have one experimental arm, and you make reference to one of the figures on there. You have one experimental arm that starts. So it's arm one against control. For the first, there's 10 periods of time within this graph. Yeah. By the way, this is the challenge of a podcast, is try to describe to people without actually showing them a graph. where arm 1 and control are enrolling equally 1-to-1. And then a new arm is added in time 3. Arm 2 is added. Arm 1 is still being used, and control is being used. And now it's 1-to-1-to-1 for control arm 1 and arm 2.
Yeah.
That continues in period 4. In period 5, arm 1 goes away. It's done enrolling patients. Arm 2 is there. Now we add arm 3, we add arm 4. Arm 5 eventually adds through the 10 periods. So you can imagine staggered arms in the trial enrolling, a common control throughout being randomized during the trial within this setting. And then the question comes down about how to make inferences about one of the arms relative to control. One of the simplest thing would probably be to just compare that arm to only the concurrent controls. And let's avoid for a second, you talk about having multiple placebos, maybe avoid that for a second, but I think that's something you importantly want to talk about. So when we're making inferences about a particular arm, do we compare that arm to the common controls that were randomized and eligible for that control at the time? Do we use other controls that have been enrolled, say, before that arm was enrolled? So you have a control patient that was randomized in the same trial. At the time, if we're talking about arm 2, it was randomized when arm 1 was there, and it was a control patient, but before arm 2 was there. So that's considered a non-concurrent control.
Right.
And so are we going to use that in some way to make inference about arm 2? Is that a reasonable setup of the problem?
Yeah, I think that's reasonable.
Okay.
There is a relationship to incomplete block designs, where typically in an incomplete block design, not every block gets every treatment. I mean, that's basically why it's incomplete. Mm-hmm. And the sort of tradition there in agricultural statistics, where they were often used, was that they had to be connected. You had to somehow be able to construct the treatment effect on the basis of a number of within block differences. However, later, I think it was Yates realized that there was some further information that was recoverable in another way if you randomize between blocks. So that's another complication. But basically, they had to be connected in order to actually make the comparison using all the information that there was. Otherwise, you were just limited to those particular blocks in which the pair of treatments you were interested in happened to be represented.
Right. And in a lot of these platform trials, you get this multiple overlapping or bridging, where when arm 2 was there, it was there with arm 1 and control. And so earlier arm 1 and control provide some potential information.
Yes.
So we could do only comparing to the concurrent controls.
Yes.
We could do as you described, where we put in a covariate for each piece of time.
Yes.
In a frequentist fixed effect, and we add in 9 degrees of freedom or something like that for time within that. What the Bayesian time machine, or the Seville paper talks about, which we'll come back to the guidance, but the FDA guidance references this paper and a couple trials it's used in, GBM Agile, for example, that it takes those units of time because they are ordered chronologically. It does a smoothing estimate of the effective time over these blocks. Coming back a little bit to the regression effect, but this smoothing over time, yes, it uses largely a smoothing spline over time, and that's the reference to the time machine. And it's done in a Bayesian way, but as you say, this could be done in a frequentist way.
The frequentist, there would be a slight a problem in a frequentist one, which some Bayesian methods could avoid, is that the polynomial you could fit would change as the number of periods were added. Obviously, very early on, you can't fit a polynomial with 4 or 5 parameters. 4 or 5 is still less than the 9 or whatever that you would use for a full frequentist fit, but earlier on you couldn't do that. So potentially you'd actually be smoothing, because as you went later on, you could actually revise some of your smoothing things. You'd actually be smoothing in a slightly different way as you went further on. Now, it's inherent to Bayesian approaches that you learn as you go along anyway. So, that's not necessarily seen as being a big deal there.
Okay, so in some of these platform trials, when you use this model, and we're making inferences about arm 2, it does use controls from earlier in their comparison to arm 1, which is also there with arm 2. So it does use these, essentially adjusting for time.
Yeah.
And we have a quote in that paper, which you put in your blog, where we say, and there's 2 parts to this I want to talk about, that largely, we're in a world right now where you hear a ton about real-world data, historical data. You could go out and get historical controls for a glioblastoma trial and use those to help make inferences in your trial you're running now.
Yeah.
That data is from a different protocol, different data. It's got a lot of things that a non-concurrent control in a platform trial doesn't suffer from. So it's the same protocol, the same data elements. All of these controls were randomized. Now, it was at a different time. So we say time is the thing that's different. You brought up that there's more than time, and I want you to maybe touch on this again, that it's not just time.
No, so let me give you an example. If you were to have a look at trials of nevirapine in HIV infection, then a lot of them were placebo-controlled. But what did that mean? It actually meant that because AZT was already approved as a treatment for HIV infection, it meant that the patients were all getting AZT, but some of them in addition got placebo to nevirapine, and some of them got nevirapine in addition. And some people would describe that as being a placebo-controlled trial. I would. Others would say, no, it's a trial of the combination therapy of nevirapine and AZT versus AZT alone. Doesn't matter. All the time in clinical trials, all the time there is background therapy. All the time standard of care is evolving. So when you said just now that the protocol is the same, yes, but the world doesn't stand still. And actually, in any particular serious disease, what you will find is all the time that your trial is running, then in that case, the world is evolving. And to return to the HIV trials, we know that people who were recruited later into the same trial and given the same treatment had better survival than those who were recruited earlier. Why was that? It was because we were learning more about the treatment of AIDS as time went on. And all the patients in those trials continued to benefit from the improvement in care that was going on. So, yes, I agree with you. It's time in a sense, but I don't agree that the fact that the same protocols are used deals with all the problems, not by a long way.
Okay, so part of it is all the problems, but the other part is some of the problems. As a comparison of external data to some extent, it's a much higher level of that, but doesn't, okay.
But it doesn't deal with all that. And then I think also there's one in which an earlier paper of yours very rightly discussed, but this later Saville paper didn't, that you discussed the fact that it would be impossible to blind all of the treatments to each other in these particular trials. Yeah. These platform trials.
Let's not move on to that yet.
Well, let's move on. We'll get onto that later then. Yeah. Okay.
Okay. So in the setting here, let's think of this as a common control. And not that there's different modes of administration of a control.
Let me ask you one other question.
Yep.
It's not the case, I don't think, but correct me if I'm wrong, that you will necessarily stick with all the centers through all the life of the platform trial. Because it's not the case in a standard parallel group trial. I've been sat on many data monitoring boards, and you find that recruitment is poor, and the sponsor says, oh, we'll enroll some new centers. So the enrolling of new centers, the dropping of new centers occurs all the time. So your differences in time are also differences between centers. And you wouldn't treat a parallel group trial as you would treat a cluster randomized trial. So therefore, this center effect is something you have to deal with. It's dealt with automatically in a standard fixed allocation trial. It's not dealt with in a platform trial.
Yeah, one of the nice things about many standing trials, the I-SPY 2 neoadjuvant breast cancer, that trial ran for 10 years, essentially it started with 20 sites, and these sites lasted for 10 years with very little changes to them. So
okay, well, that's
remarkable.
Benefits,
right, one of the benefits of the platform becomes almost this learning healthcare thing to that. Now, you're right that there is some variation to that. In a number of trials, we do try to adjust for center site. One of the things about this, though, as you mentioned, time, and maybe this is semantics about whether time includes the evolution of background care and other aspects to it. But we also empirically have these observations on the control arm over these 10 periods. So we empirically see that outcomes are getting better over time. Now, there's absolutely the assumption of additivity of that across arms. So within the model, that an additive effect, whether this is a hazard ratio, whether this is a responder analysis, empirically we can see whether or not this is happening during the course of the trial.
Yeah, I have no problem with that. And in any case, I don't really regard the additivity assumption as being particularly important. I'm less concerned about that. I regard that as being a treatment by time interaction. What I'm really interested in is the main effect of time. And I think the main effect of time is something that can be underestimated if we're not careful. But essentially, you're replacing a model with many parameters with the one with fewer parameters. That's not necessarily an unreasonable thing to do, but I'm not sure that everybody who's involved in a DSMB understands exactly what's going on. And in particular, the question is, how should data that they use for monitoring be presented to them?
Yeah. So a huge issue then is during the course of the trial, the DSMB is reviewing that there's this model that's making time adjustments, and do they just accept that, hey, the model's got this, don't worry about it, or are they able to view this, are they able to see it? Hugely important. Let me come back to what you said about other type of trial, and I'll make reference to the Healy ALS trial, which has the components that you just brought up. In many trials, what happens is there isn't a common control. Oncology, for example, there's a standard of care, and patients generally aren't even blinded in oncology trials because of the intensity of the treatments. But in the ALS platform trial, a patient is randomized to, say, drug A, B, C, and then they're further randomized 3-to-1 to its placebo or its active.
Mm-hmm.
So at any time, if there's A, B, C enrolling in the trial, patients are being randomized to A's placebo, B's placebos, or C's placebos. They are not blinded to A, B, or C. So if one of them is 3 a day oral pill and one of them is a subcutaneous shot, they're not given the blinding subcutaneous, they're only given the mode of administration of the drug that they're on. So at any one time, we have modes of administration of placebo. So not only do we have controls that had ALS that were enrolled slightly before the arm came on, but we also have placebos at the same time that are given different modes of administration of a placebo across the different arms.
Yeah.
And I think you wanted to talk about that.
Yes. I mean, I think I've referred to such trials a long time ago. I'm trying to remember when it was, as veiled. If you don't know what treatment you're getting, but you know some of the treatments you're not getting, then in that case, it's not fully blind. It's sort of veiled in that particular way. Oh, I use the term veiled as being, partly obstructed. Just have a look.
A good term. Yep.
Yeah. I think it might have been, 2004, I think it was.
Okay.
And I was thinking of a particular trial that we had run at Ciba-Geigy where we had 2 patches of hormone replacement therapy, a high dose and a low dose. And that meant the patches were of different sizes. So the only way you could have blinded the patients would have been by giving them 2 patches, a large patch and a small patch. One of them would have been, let's say, active, and one of them would have been placebo, but they wouldn't know which. And then you could maybe have had a placebo group with 2 patches, which were both placebos. And then basically a patient who's being given the highest dose knows that they're not being given the lowest dose. So if expectation leads them to report side effects, because they say, wow, I'm getting a high dose of hormones, this could be a problem, I don't feel so well, and they report it, you only control for that by comparing them to their own placebo. If you compare them to the pool placebo group, then in that case, you don't actually control for this. Another study I was involved in was a target study where lumiricoxib was treatment for rheumatism, osteoarthritis, was compared to naproxen twice daily or ibuprofen 3 times daily. And again, it was awkward to blind things. So basically, you had a sub-study, which was lumiricoxib versus naproxen, and another sub-study, which was ibuprofen versus lumiricoxib. And on the Data Safety Monitoring Board, we had to take great care to make sure we only looked at the control patients from the same sub-study, because the results in the 2 sub-studies were just not comparable. So you would have got a bias. You would have been biased in actually doing the monitoring if you hadn't split them in sub-studies. We had to do essentially treat them as 2 separate trials.
So in that case, the outcome sounds like it was osteoarthritis pain, for example?
Well, yes, although to be honest, the trial was also looking at, because the second generation of COX inhibitors, the sort of COX-2 inhibitors, were supposed to be better in terms of gastric side effects. So one of the things one was looking at was gastric side effects, but also there was a question mark over cardiotoxicity. So one of the other things one was looking at was cardiovascular side effects. But the problem was that the 2 sub-studies were not comparable. So same protocol. The only thing that was different in the protocol was essentially the treatments that were given.
So do you think that that's context specific? So for example, in the ALS trial, we have at the same time patients that are given different placebo by different randomization. Endpoints are functional rating scales. They are mortality combined together. By the way, we have randomized comparisons of those different controls in the trial. So if you're making inference about drug A and it has its placebos, I would shudder to ignore placebo B and placebo C. First of all, we have randomized comparison, and additively, are they responding differently? But in some diseases, and we talked about early on the regression of the mean and what is a placebo effect, and much of the placebo effect is protocol driven and not thought that I'm taking 2 pills a day instead of 3 pills a day are going to affect the time of mortality in an ALS trial.
Yes, that's true. And I don't necessarily argue against using let's say a model with fewer parameters. I'm not saying that that's the case. I'm just saying that these particular issues are not necessarily discussed. For example, to return to the time machine, in your particular Saville et al. Paper, you're looking at comparing a model which essentially has got 9 parameters for time with your spline model, which has got rather fewer. And so basically, if everything's okay, then you're going to do better with the model which has got fewer parameters, no question. I mean, if you could ignore time altogether, you'd be even better in the naive pool model, but that's right. But nobody's going to go that far. But actually, I could argue there are not 10 groups, there are 24 groups. If you look at the combination of time period and control, taking into account the blinding thing, you don't end up with 10 groups, you end up with 24. And in that case, the degree of adjustment is going to penalize you a lot more. So it's not even true that I forget what you call it, the time categorical?
Time categorical, where, fixed effect.
Time categorical, fixed effect. It's not even clear that the time categorical effect gets rid of all the biases. Actually, if you really believe in concurrent control, you have to have 24 groups, not 10. So you can still make the connection. There's still a sort of connection you can probably make, but nevertheless, it's going to be a lot more difficult.
Right. Right. And get that, the fully parameterized scenario in that. And is there a different effect of the placebos, and does that vary over time? Starts to get hard to imagine in all of that, but that has ramifications in trial design. So when we're designing one of these trials and we do the amount of randomization to a control in that, forcing the fully parameterized model means we have less investigational arms. We have to enroll more patients to a placebo who have ALS for 12 months, for example. Has massive implications to the design. So hence the statistician plays this interesting role where it's the concern about 24 parameters, as you say. How much modeling do we do? How much are we willing to do? And the ramifications it has on the disease and the global state of treatment.
Yeah. Yes, I as I say, I'm not arguing it always. Applied statistics is a bias variance trade-off. It's one of the first things that you learn, and you can't always come down and insist, well, I want the unbiased solution, because it really depends on how complex a model you envisage, what on earth that would mean. And so a certain degree of bias is accepted by everybody. I'm not arguing against that. But I think there are some things which are happening with adaptive designs. First of all, I think that the claim for efficiency that was made has been somewhat misleading. I think there is a big benefit in efficiency. I think it's mainly organizational. I think it's not so much being able to use the same controls over and over again, because, as you've already argued, there's actually less information in that than one might think, because of the possibility of adjusting for time effects. As soon as you start doing that, then you find the standard errors will go up.
But it's sort of compared to what? Compared to only looking at the concurrent controls, there can be huge advantages.
As I say,
building that model.
They're not as great as sometimes claimed.
Okay. Okay.
I mean, you can see that from some of the Bayesian work on using historical controls, which I like very much. I'm thinking of the sort of work that Heinz Schmiedli and Beat Neuschwander and people like that, based at Novartis, have used for using historical data. And we've done a similar thing in a frequentist model. And what you find is you can identify, in one of our cases, we identified 1,200 historical patients. But when you looked at the between-study variation, it was equivalent to having, optimistically, 50. So 1,200, you thought, wow, I'm rich. But actually, when you had a look at between-study variation, because you're using historical data, then in that case, the information was not nearly as great. Now, I'm not saying something as drastic as that happens with platform trials, but some of the discourse has gone in the simplistic way of saying, oh, and we can use all this control data. Well, it's not quite as simple as that. But I think the organizational side is a great saving. And that was also seen during the COVID epidemic, in which organizing oneself to drop and add arms and so forth quickly with a minimal amount of administrative fuss was very important.
So you touched on a larger topic sometime, scientific hype and actual reality a little bit. You talked about adaptive designs, but largely platform trials. There's been the new FDA guidance is out, draft guidance on Bayesian statistics. ICH E20 draft was out, which talks about adaptive designs largely. Do you have, what is your thoughts on all of that and the movement towards some Bayesian to adaptive designs?
Well, I mean, decision analysis teaches you, and I'm not denying it, that the option to change things is always valuable. So from that point of view, you can't argue against flexibility. The option is not always as great as some people think. I'm slightly annoyed about all of this because there are other simpler things that the FDA could have been doing a long, long time ago, which would have made a much bigger difference. One of them would be banning dichotomies. An extraordinary number of clinical trials still use information-destroying dichotomies. And we know that as soon as you do that, on the best of cases, your sample size increases by the necessary sample size increases by 50%, but it can easily double, treble if you get the cut points wrong, if you get bad ones. So that's one particular point. The second thing was using covariates, which one could have been using in a linear model sense years ago. Now the FDA has gone up on some ridiculous covariate hunt in terms of estimands and so forth. All of this is really of minor importance. The important thing was to use covariates and model, and we could have been doing that a long time ago, and we weren't. I've even turned up at particular meetings in which the head of a particular section, statistics section of the FDA said proudly, "We don't do modeling." And I think, gosh, how can a statistician say such a thing and be proud of it? It's unbelievable. So
I'm fully on board with you on both of those points. The dichotomy is just it's mind blowing that we do
insane, absolutely insane. You replace the whole Kaplan-Meier curve that you could have, all that information. You replace it by just 2 points on the curve, which is response rate or the death rate or whatever the rate is at 2 years, rather than having the whole thing that there is there.
Right.
And as Keynes said, in the long run, we're all dead. So,
So you had touched on something else that I think is important, and I want to make sure you were able to talk about it. Is a DSMB in a more complicated trial, within a platform trial, time adjustments going on, and the role of or the challenges of that?
Yeah. So, I think that's challenging because what you're typically looking at is you're looking at all sorts of side effects, potential side effects. Let's call them adverse events, without really knowing whether they're causal or not. And it's very unlikely that you will have the machinery for doing the time adjustments for all these things. So you're actually having to make some sort of judgment just by looking at raw data, where ideally you would want controlled data. You'd like to be comparing like with like in order to do that. So this is one of the problems, certainly.
Yeah. So I do think it's in these more complicated trials where you have human oversight, diving into what the model does know and doesn't know. The model knows certain things, and maybe there's a trust there, but there's lots of other things that it's critical to be able to do this well with the DSMB for sure.
Yeah.
You said something as we were coming on, and I wonder, I think it'd be valuable for everybody to talk about, but you described this evolution. And largely my question is to you, looking forward, what things do you think are important in clinical trial science? You've seen a good deal. We talked about you've been a statistician, you've been a professor, you've been at a pharmaceutical company, lots of roles in this. So thinking about things going forward where we are. One of them you said, which I thought was really interesting, was the difference between eras in which we had private data and public analyses to public data and private analyses. I'd love for you to tell our listeners about that.
Well, I suppose even though I'm a frequentist, I usually do frequentist analyses, and even though I sort of believe in the value of pre-specified analysis, I sort of wonder, well, especially when we look at things like multiplicity, is it reasonable that just because a group of us got together on a particular day and we decided on this particularly complicated scheme for adjusting endpoints, that the whole of scientific posterity is now condemned to use this particular scheme that we chose? Of course, the Bayesian answer would be, well, they're not, because people are not required to think alike. They start out with different prior distributions. They have different values. And the sort of depressing end result of that is that we end up sharing data. There's no analysis. There's still some value in trust. I often say that you should think of the purposes of a protocol in terms of the 5 vowels. A for anticipation, it's your thought experiment. E for ethics, it's the way in which you explore the ethical problems that could arise with the trial. I for inference, which is what you and I are interested in. O for organization, and U for utmost good faith. His utmost good faith was important. If we're going to share data, then we have to know how did the data arise, what was done before we saw them, which comes back to this point we were discussing earlier.
Yeah.
So I think that we're moving towards an era in which the data will be available on the web. We're going to have all sorts of problems with anonymizing, but then we will have to sort of adapt a quotation of Chairman Mao's, let 1,000 analyses flourish, So we're going to see lots and lots of different analyses, and the problem of multiplicity will enter a new world. And maybe it's not so quite much hidden data, it's analyses missing not at random we should worry about. Only the interesting ones will be reported. The dozens and dozens of boring ones will not make it. So I think that's an issue.
Yeah.
And relates to that, I think, is also the idea of the evidence from a study, which was always rather suspect from a Bayesian point of view, because it would depend on your prior distribution as to how evidential the study was, is also coming under scrutiny. And you can see that with your adaptive design. If you have a look, you'll see that the information continues to accrue for treatment number 1, even though it's been abandoned in the particular design you're looking at, because what's happening is we continue to have control. So although we learn nothing more directly about treatment number 1, indirectly we learn something about it because of the other arms. So there is no fixed evidence from a particular study. It's all relative.
Yeah. And that actually was absolutely, in the I-SPY 2 trial over 10 years, we had 27 arms. And the inferences about arm 1 that was in there continued to change. Now, it was very, very small, but it did continue to change as the data accrued.
That's what you would expect. Yeah.
Right. Do you find value? A question out of nowhere. Do you find value, since you rail against the dichotomization of endpoints? Do you have a similar frustration with the dichotomization of a trial as success or failure? And does a Bayesian play a role in quantifying evidence, perhaps above and beyond frequentist in those trials?
Well, I think that there's a sense in which, at the point at which you have to make a decision, things are binary. So you do have to make a decision for a given patient as to whether to use one treatment or another. In theory, what you could say is, well, we're going to delegate the decision-making away from the FDA. What the FDA will do instead is the FDA will say that these data have a seal of approval. They are valuable data that you can use to make your own decision. Now it's over to you, the doctor, and the patient to make the decision. And a long time ago, Jürgen Hilden, a very good Danish statistician who was interested in utility theory, he proposed this. He actually said that what you should do is you should produce an analysis of all the various outcomes, all the things that might matter to a patient, and then every patient could look at them together and they could do the trade-off themselves. They could decide whether they would. So eventually, there would have to be a binary decision. The patient's going to have to decide to take pill A or pill B or something else. But it doesn't mean that you have to think of a trial in that particular way. Provide information. I can see all sorts of difficulties in making this a way in which society will behave, but I wouldn't necessarily argue against it. I think, I think it's also, by the way, I think relates to this is a misunderstanding about clinically relevant differences, delta. The delta for me is not what you expect the drug to do. It's essentially some way in which you scale the information. Because what you want in a trial is you want the trial to provide a valuable amount of information. And that means that the data precision, essentially the standard error divided by some function of n, so standard deviation divided by some function, square root function of n or whatever, that should be some multiple of what you consider an important quantum of information to be. It's that particular ratio that you're targeting, and the clinical relevant difference is a sort of way scaling that. So yeah, I think trials they're not failures or successes. The trials provide a certain amount of information, and then,
Awesome. So, any other closing comments, Stephen?
No, apart from giving my best regards to Don.
Well.
No, I don't think so. I mean, I think apart from, I would say that people should think about concurrent control. It's not the be all and the end all, but it does all sorts of things for you. And also blinding. If you can run a double-blind randomized trial, that also cures all sorts of things. If you're not careful, you're liable to overlook things which would be impossible if the trial is randomized and double-blind. For instance, in a trial of a vaccine, you might say, well, the people we're going to vaccinate, they can come to the center to be vaccinated. We can't run the trial double-blind, so there's no point calling the control people in. We'll get nurses to go and take blood samples from them to see if they're seropositive or negative. And already what you find is that now the blood samples are being handled differently. And maybe they're being sent off at a different time in a different lab. If it's the same lab, then we know that assays vary over time. And so without really realizing it, the measurement process itself is introduced a bias simply because the thing is not randomized double-blind. If it's randomized double-blind, it's impossible. There is no way that you can correlate the taking of any sort of measurement with the allocation of either treatment, because it's random and nobody knows what the treatment is.
Agreed. Well, thank you so much. Thank you for your blogs. Thank you
Pleasure's
for
mine. Okay.
Yep. Thank you for the pictures of your hikes, and enjoy your hike tomorrow. I know you've been in a lot of interims, but thanks for joining us here. And for everybody, till next time, we'll be here in the interim.
Okay. Yeah. Thanks. Bye.
Thank you.